Um, I should start with this slide, in fact, because this is the work largely led in the last 7-8 years by Brian Graham. Brian is a pulmonologist, a physician scientist. And with Raul and Claudia, he is largely responsible for this work. I should also highlight it is a multidisciplinary work. Like I mentioned to several people, I try to avoid missteps when I get into a new field that this was starting in 2008. And this would not have happened without the help by Thomas Wynn. Many of you know Tom. He's an expert in TH2 and fibrosis. And it is work that we have collaborated very closely with people in Brazil to have the relevance for the human. So I know that many of you are clinicians, are seeing patients, but I'm going to highlight very key data in an animal model, because this is the way that we can start making inroads that addresses many questions that Mark Krasno had outlined in the previous sessions. So Mark asked, what do we know about cause-effect relationship of inflammation and the disease? And you already have seen many slides of association. This is another one, but this came out from me in 2012. I'm the pathologist involved in a large network of PH tissue. Marlene is also directly involved in collecting these lungs, so we reviewed in excess of 60 lungs, including scleroderma-associated PH, IPAH, some cases of familial or hereditary as well. You have seen this picture showing clustering of inflammatory cells, but this was the first time that actually the statistics worked out correlating what we call the perivascular inflammation score that was done semi-quantitatively, but pretty much reasonably quantitative. It's less opinion, but more quantitation. And with mean pulmonary artery pressure, but also with the remodeling. And by using this large cohort that was collected in the last 4 or 5 years, this means all of these patients, they were terminal in terms of disease, they underwent lung explant, and they were all being treated by the drugs that Mark outlined that are currently used. So nevertheless, even being on these therapies and eventually evolving into end-stage lung disease, you can see that the correlation holds true. So it's like what I said before. It doesn't fulfill all the Cox postulate, but it's a very compelling argument that even with the new drugs or the current drugs, there is a very strong association. So this is the only human tissues data that I'm going to show you, because I'm going to move into schistosomiasis-induced PH. I'm going to highlight for you why is it relevant, significant, and important. One is that there are 200 million people infected by this parasite. It's predominantly in temperate areas, namely China and Brazil are very high, but there is also cases in Sudan and north of Africa. So out of these 200 million people, between 1 to 10%, the most frequent cause of PH worldwide is going to be because of schistosomiasis. We can talk about IPAH, an infrequent disease, a paradigmatic disease that we learned so much, but in reality, we're talking about worldwide issue of related to pulmonary hypertension. This is the disease that we should have been talking about. And despite Tom's inroads in understanding how the immunopathogenesis of schistosomiasis affects the lung, but mostly in the liver, in fact, we knew very little when we started working with Tom in 2008 about the mechanisms underlying this disease. So how it happens and why is it so prevalent? It has a very relatively straightforward cycle, and it's a phenomenal disease in terms of inducing fibrosis and TH2. And here is the egg. This is an individual. You can see that there is hepatosplenomegaly. And the reason that this happens, I'm going to show you in the subsequent slide, is there is a process of fibrosis in the liver, and then there is portal hypertension leading to a massive splenomegaly. In these individuals, they get infected because during in the middle of the day, they go to wash things and do the work in the riverbanks, and there is a snail which houses the intermediate form of the parasite. It releases a cercaria, and the cercaria is actually an amazing little organism which can break through the skin in the water and then penetrate because it has hydrolytic enzymes. And then once it gets into the venous system, circulates out, into the lung, breaks through the capillary surface, and then gets eventually ingested into the gastrointestinal tract. And then the male and the female nest in the large intestine and release eggs that are going to eventually flow into the liver. So what happens in terms of the pulmonary vascular disease? You guys saw in the classification, there were 2 slides already. Schistosomiasis has been introduced in the last classification, meeting in Nice as the last form of PAH. And here is a casting of the pulmonary arteries, and this was provided by a professor of pathology in the state of Bahia in Brazil when I was there visiting, showing that these casts are completely obliterated. So this was a plastic casting. In all of these branches, instead of progressing towards the periphery, you'll see that they are pretty much occluded. They have plexiform lesions, which you already have seen before. And what is really compelling in this disease, you have a specific agent that is the egg nesting around the blood vessels and then leading to the vasculopathy. So rather than talking about things that we don't know how the cause-effect relationship establishes, we have something that we know the culprit. We know the agent. And therefore, we can build a paradigm based on that because we have initiation. We don't know what initiates IPAH. We don't know what initiates scleroderma, associated PH, and, for that matter, hypoxia as well, but Kurt is going to make an argument that is otherwise. But how we model that in the lab, we have to move into the animal model. I know that many of you is going to be turned off by going to the animals, but we have to start understanding somewhere. In this case, we get the snails. They read the New York Times, and during the times in the lab, they actually release the cercaria. We have eggs as well. So we do a modeling system that is abbreviated because usually the normal course of the mouse, like in the human, takes a long time to develop. In terms of the mouse, 4 months, 2 years lifespan. In the human, probably recurrent infections and recurrent nesting of the eggs in the pulmonary circulation. But what we developed in the lab, largely Brian's work, that we sensitize intraperitoneally using the eggs, and eventually 2 weeks later, we go and we inject the eggs now via tail vein, and it's 5,000 eggs. If we do only that, nothing happens to the animal. The animal, pretty much disregards the challenge. But if you do the double hit, we can now model in a 3 weeks' time, which is manageable, a very complex disease. So what we learn out of that, just for the sake of introduction, yes, it causes pulmonary vascular remodeling. You guys can appreciate that here, but causes, differently from the Gabriel Grunick's paper in JXMed in 2008, if I'm correct, it causes an increase in pulmonary artery pressures. That's pretty reasonable in terms of even a mouse response. And here is a parameters of remodeling, particularly media remodeling, right ventricular remodeling. So this has been published just to show that the model is relatively robust and reproducible. So we went systematically. I know that, again, this kind of linear paradigms may turn you off because every single time the disease is much more complex. However, we need to start somewhere. So if you want to get from point A, Denver, to Stanford, you have to go via some routes. You have many routes, many choices. But the one that we decided to pursue in the last 4 or 5 years, that we start with the schisto, and I put here the antigen because this is one of the holy grails that we need to start addressing because we have a complex organism. And if we can define a little bit more on the antigens, we can actually dissect the process even in a larger, more detailed manner. It engages a prototypic TH2 inflammation with these 2 cytokines. Only one is not sufficient, it's both. Activates p-STAT6, leads to TGF-beta via SMAD3. There are different SMADs that signal TGF-beta to 3, and 4. In this case, it's 3, we know, because of mice that have been deleted for SMAD3, and creates the vascular remodeling and eventually pH in the mouse or PAH in the human. So let me jump here. So what is the next question? So this we already have shown. So we decided to focus in the last year, year and a half, in a couple of elements. Are the cells that are producing IL-4, IL-13 critically important, namely which cells? And I'm not going to show you data because this just came out. I was told it is already in the website, in the Blue Journal, that IL-4, IL-13 come predominantly for the CD4 cells. So you deplete the CD4 cells, we cut the proximal component of this pathway. And I'm not going to discuss the data. Just to summarize that we need both. Just one of them is not sufficient. That p-STAT6 is involved because we can block it, and we block the downstream effects of remodeling and pulmonary hypertension in the mouse. We had already shown that TGF-beta SMAD3 signaling was important. This is a circulation paper. And what I would like to highlight is, in the context of this conference, is because we have the agent, we can time the process, and we can measure endpoints in a very robust manner, very detailed manner, to define this population of macrophage monocytes that is the linchpin to start defining what is a bad guy, because Mark is absolutely correct. I think that we get on the bandwagon of calling everything bad. It's profiling. You don't like profiling people? Don't profile immune systems either. There are good guys and bad guys. And the other thing is that, like people, some good guys can become bad guys, and some bad guys can become good guys. So this summarizes that really a very important element that we still don't understand. Without that, I think that trying therapies is probably going to be premature. And then I'm going to focus on this molecule because this is a linchpin out of this population to activate the TGF-beta. And this is in the context, of course, of pulmonary vascular remodeling. Oh, gosh. I'm sorry, because this is a lost in translation, the apple. That's, anyway. So what I had here is TGF-beta. I never put black on blue background, please. As a pathologist, I'm colorblind to some extent, but not to this degree. So anyway, what I was trying to show here is that the TGF-beta resides in the lung, in the matrix. And TGF-beta is such an important cytokine developmentally and also in terms of adaptive responses that is actually anchored in the matrix that was supposed to be this little thing here into a latent associated protein that pretty much anchors into the fibrillin network of the lung, just in case. And this keeps the TGF-beta kind of a pull on an inactive state. So a couple of critical steps involves the release of this anchoring by the latent associated protein. So comes a protein that is in the very large protein, that's thrombospondin 1, that's in the blood. And it's a very complex protein with multiple functions. But in these repeat domains is a sequence of peptides that I'm going to show in the subsequent slide that is capable to disanchor or de-release TGF-beta from the latent associated protein. So it's a peptide-peptide interaction that releases TGF-beta and allows it to be functional. So how can we manipulate that? My gosh, it's really, it wants to be in the background. But anyway, you can manipulate that because if you go into the peptide sequence, this tetrapeptide is critically important to dislodge the TGF-beta from the LAP or latent associated peptides. So now you guys probably understand how we can manipulate that, which was done largely in the 90s. You can take this peptide, if you put it in a biological system, releases latent from the TGF-beta, and the TGF-beta becomes active. Conversely, if you take the peptide from the latent, and this case came out okay. So you take the latent, the peptide that is in the latent associated protein, LSKL, and you give it to the system, is going to block and quench the activity of thrombospondin. So the experiment is straightforward now. Of course, it takes years to be able to be straightforward. But you can see here is that question number 1, is TSP1 upregulated in schistosomal-induced PH in the mouse? The answer is yes. This is by RNA-seq data we are documenting. We have already immunofluorescence, immunohistochemistry, and this also got completely shuffled. But the important thing here is that if we use the peptide that is a loss of function, that blocks TSP1-induced TGF-beta activation, you improve the hemodynamics. And the remodeling is the same thing. I'm just highlighting the key data for you because of the sake of time. And how we control this, we have a scrambled peptide, very similar, but with one substitution, or replacement or swapping, and you can see that's beneficial. And I'm not showing you all the controls, but I'm showing the gist of the data, which is absolutely probably dramatic. Because this takes me to the next step before I wrap up. You can see here, this is the wild type. They are infected. I'm not showing you the controls. The controls would be over here. There is increase in pH. If we take a wild type TSP1 bone marrow and put it in the reconstituted mouse, induces pH like the wild type. If we take a bone marrow from a TSP1 knockout, improves pH. So clearly the TSP1 is coming from the bone marrow compartment. The question that Mark alluded before, is the alveolar macrophage a good or bad? Is the bone marrow contributing? In our data here, and probably in a lot of other model systems, probably the bone marrow is the bad compartment in terms of inducing a release of a culprit cell that can activate TSP1. So going back to data that, we use the same approach that several years back Kurt used. We depleted the macrophage monocyte population with clodronate, and indeed there was a decrease in pulmonary artery pressures, in this case RFSBP, but no impact in terms of remodeling. So this actually is concordant that something is coming from the bone marrow, probably monocytic macrophage population, that is possibly activating TSP1. So how we go into defining it? So we take the lungs now, and we flow sort, and this would not be possible again without the help of Bill Jensen, who is an expert in macrophage biology, working with Peter Hansen in the National Jewish in Denver, and he assisted us with that. You can see here is the control, and here is the schisto-infected wild-type mice, and you have this population here that has this phenotype. Is CD11C intermediate, CD11B positive, CD64 intense, MERTK, that's also a marker for macrophages, monocytes, is part of this population here. But probably the most compelling argument that these cells are not just inducing another cell probably to make the TSP1 is that these cells are the ones that have, here's the infected cells that have TSP1. So we probe TSP because TSP is soluble. You cannot just do it in cell surface. So here is permeabilized cells, and this is cytoplasmic staining. So this population of macrophages, we don't know if they are sufficient to cause a disease, but at least now we have a phenotypic characterization for some using these markers that allows us now to monitor what is the transcriptomic profiling, very much in line what Mark suggested when he came to Denver several months back, and it's based of his science paper, right? Nature paper. So to finalize, and it is a very abbreviated, I didn't go over tons of data. It probably is not worth bothering with additional information, but probably what we are working with is that, again, I cannot emphasize enough that it is critically important in the field that we have a trigger agent. Because if you don't have a trigger agent that's specific in terms of immune response, we cannot map how the immune response has to be harnessed in order to devise a treatment. Secondly, we have probably a very, very complex processing of antigens. And which antigens is our next step? Because I think this is the next critical experiments that we need to do to define which antigens are the ones, because it allows us to dissect even further which cells are mediating the priming of T cells, predominantly CD4 cells, to release IL-4, IL-13. Here is absolutely a critical cell in our model system. The macrophage or monocyte, which we are trying to start to phenotype and we have a handle now, is critically important to activate TGF-beta, which is essential for the remodeling. And this is done via TSP1, and then this causes the disease. Why is it important? We had a question, a very good question, about antifibrotic therapies. Why TGF-beta targeting is still not in the clinic? Because it has enormous toxicity. Shuts down the kidney, causes autoimmune disease because it's a suppressant for T cells. So the only way that the field is moving forward, very much what Dean Shepard is doing in fibrosis at UCSF, you go through the mechanism by which TGF-beta is activated in the context of the disease that you're studying. And this is the reason that we believe that TSP1, which actually was investigated in the 90s but never linked very clearly to a specific phenotype or a disease, is probably critically important, at least in this system. By the way, I would like just to finish saying that every single thing that we do with the mouse, we validated the human, at least descriptively. So I wouldn't be presenting this data if it, at least without the mechanistic component, because we cannot do it in the human. But all the elements that I showed you in this paradigm, they have been validated in the human lungs that we have access from Brazil. Thanks so much. Yes, Roger.
Could you comment on the unique nature of the parasitic antigen in regulating macrophage phenotype and the role of the
that's a wonderful question.
compared to other pH diseases?
Can I just correct you? I think that the work that Brian and I have done, let's give credit to the person who deserves it. And I think that you're absolutely right. And I think that when we started this work, this was something that I had in mind, my credit, but I just didn't know how to do it because we needed to have the mechanistic steps and the phenotype before we would be able to go back to the agent. So we really need to start addressing it because your point is absolutely correct. So if we're the same, at least in terms of schisto, if it's a specific pathway, there has to be specific antigens. Could be multiple antigens. But definitely we need to be able to reproduce the key steps with the antigens rather than the egg. To propose that it is an immune-mediated process. Because could be occlusive, could be injury, could be cell death, could be everything. But I think that this is what is missed. But I think that you would agree with me that without mapping this pathway downstream, we would not be in position to start dissecting. It would be completely premature. We need to reproduce with the eggs rather than, but you're absolutely right. Mark?
Two points really quick. I don't know if this is on. So intimal fibrosis then is a feature of this mouse model?
Yeah, I think that we have to qualify. The intima in the mouse, in the vast majority of circumstances, is very wimpy, in general. So the mouse does not remodel with intima remodeling. It's largely media. So the way that we see intima increasing the thickness is because of our measuring approach. It's reasonably sensitive to be able to see thickening, but it's not to the point that you would claim that it occludes the vascular lumen.
Okay, so partly addressing a question that was asked earlier, is it spironolactone, which is being used in pulmonary hypertension, has some impact, I think, on vascular intimal fibrosis, and would that be something that could be tried in your model? The second thing that I want to ask quickly is in humans that get schistosomiasis but don't get pulmonary hypertension, getting back to Mark's question, do you think that genetics, the interplay of genetics and the deposition of the egg matters, and can you investigate that in the mouse?
Yeah, that two really good questions. I cannot answer the first one, spironolactone, because this model system doesn't involve so much intima. The question becomes, so why is it relevant? And here's another discussion. How much is media and how much is intima, which comes first? My point of view after all these years in the field is that both dance together. So you have to have both. This is my point of view. If you can abrogate at least one of the two partners, the dancing stops. Then the second question is, in terms of the genetics, it's a great question because unfortunately I don't have quality specimens that we can do clonality studies, or we can do genetic studies to see if there is mutations like Michaela Aldred has shown in IPAH in terms of genetic instability, which you guys probably are aware now. It's another hot area. I think Marlene also studies that in relationship to BMP, which I think that's highly relevant. So I think that it is a central part that we still have not addressed. And the only limitation that I have is quality of lungs that come from Brazil to be able to do these more detailed studies. They never passed our quality control of the DNA, unfortunately.
So, hi. Given that schistosomiasis is a parasitic infection, is there any evidence that eosinophils play any role?
That's a great question. The eosinophils are also present, and we have not. I think the answer is no. The eosinophils don't play a role, but I think the evidence comes from Tom Wynn with animals that were eosinophil depleted, and they still had the liver disease. But we have not directly addressed ourselves the importance. The thing is that you get guilty by association because it's downstream from IL-4, IL-13, IL-5 driving the eels to accumulate. So when you block proximally, the eels are going to go away. But I think that the evidence is that eels are not important.
I'm interested in this potential switch from a regulated immune response to a dysfunctional immune response and causing vascular disease. And I think your model offers something here, because if you said if you give one shot of 5,000 over, the animal doesn't develop disease, but if you give 2 shots, it does. So have you looked at the immune milieu after 5,000 eggs?
We did. We did. There is no pulmonary vascular remodeling, and there is no nothing happens in terms of pulmonary vessels. If you sensitize without challenging, nothing happens. So clearly it's the combination of the two. And I think that the reason why, let's go back to mechanisms, because I think this is really, I cannot emphasize, although I'm a pathologist, but I have to say that without the mechanism, you would be lost. So the issue here is because we have the monocytic cell population that's popping up, something is, by combining the two events, is making these cells to come out and go to the lung. And I think that this gives us a better understanding about antigen load, which antigen, Roger's question, and why it takes a certain amount of time to drive the bad cell population to mediate the damage. So I think that, again, I think that this is, this we found a couple of months back. So now it's so important because it gives us a chance to start answering the question why you need to this 2-hit process. Is the natural infection that takes 5 months engaging this population, but in a smaller turnaround? And of course, the question is that, can you find the same population in the human tissue? Marlene.
Yeah, Rubin, to what extent do you think the microbiome comes into play here? Excuse me, I'm thinking of Skip Virgin's work that the IL-4 response can be generated by different microbial environments. And
It's tough. I can be, dismissive by saying, microbiome plays a role in everything. But it's probably correct. I just don't understand.
Have you seen different strains or different
not
effects,
in terms of,
variability?
I cannot, to be honest with you, I don't know how to answer. Because, Skip Virgin work relates to virome, right? Yeah. And I think that the pH field, particularly in terms of autoimmune disease, by the way, this process occurs, I need to mention that, without TH17. So this thing about TH17 being obligatory, it's not correct. By the way, IL-6 here is protective. It's not detrimental. So this brings the point about phenotyping the patients that we are going to start putting therapies in. But then going back to your point is that if we are thinking about a particularly collagen vascular disease, which I believe the Schisto can address some shared paradigms, it clearly involves the microbiome because this can drive the TH17 versus Tregs. And I think that we need to start understanding that. But I don't think that I don't know of any work that has been done in terms of pH. And it's not trivial. I have spent 2 years working in terms of cigarette smoke, induced lung disease and microbiome, and it is incredibly complicated. So it is not easy to have a hold of understanding, but you're absolutely correct. If you're thinking about an autoimmune dysregulated function, it could be important in how to mold the TH17 in appropriate setting or Tregs in the appropriate setting as well.